Hello, welcome again in this continued lecture. In this lecture, we are going to study chemical and physical properties of amino acids. Whenever we have to study physical and chemical properties, as the amino acid contains two major groups. One is the amino acid, that is amino group, NH2 group, which is always acting as a base, while COH group, that is carboxylic acid group. It means that it shows properties of both, that is base as well as acid. Amino acids are the building blocks of proteins, and each has distinct chemical and physical properties. The same property are always reflecting to determine protein fold and their functions, and also provide the specific key to breakdown of various proteins into amino acids. So whenever the properties are considered, see its basic structure, that is all amino acids except proline. The common structure containing R group as a side chain and NH2 group working as a base, carboxylic acid group act as a acid. Next, general formula is same again, NH2CHR COH, where R is the side chain. Now see the important chemical properties of amino acids. No doubt, most important chemical property of amino acid is amino acids are amphoteric in nature. Let's see what are the amphoteric nature. As amino acid contains both groups, that is NH2 group acting as a base, COH group acting as acid. It shows reactions of acid as well as reactions of base, and hence such kind of nature showing acid base both properties in the same structure, the nature is known as amphoteric. This is the first thing. Then second important thing is that it exists in the form of Zwiter, which is neutral in nature. Now this is second very important property, that is amino acids are exist in the form of Zwiter ion, that is amino acid group exist in the form of NH3 plus positively charged NH3 group and COH group with COO minus group, that is negatively charged ion. Means same molecule contains positively charged cation as well as negatively charged carboxylic acid anion. Hence, it exists in the form of Zwiter ion, and that's why it is having neutral in the solution, neither acting as a base, neither acting as acid. So this is second important chemical property, that is Zwiter ion formation. Third, isoelectric point, Pi. This isoelectric point is a special property shown by various kinds of alpha amino acid, and same will be we have to see in details last time of the particular video. So isoelectric point is the pH at which amino acid having no net charge, that is either positively charged or negatively charged, and this is the special property. Fourth important is that buffering capacity. Alpha amino acids are having buffering capacity, that is resist amino acids resist change in pH near their pK values. By adding some acids or base, it forms a buffer and it opposes to change the pH of solution. Such kind of property is known as buffering capacity, and this buffering capacity arises again due to formation of Zwiter ion form. So this is a fourth important property. Then fifth one, amino acids are having tendency to form peptide bond. What is peptide bond? That is, peptide bond is nothing but amino group of one carboxylic amino acid combines with carboxylic acid group of second carboxylic acid group or amino acid group and gives formation of peptide bond having structure that is CONH. And this repetition of polypeptide type of bonding is found in the polypeptides so as to form various proteins. How the proteins are developed? Number of alpha amino acids combines together to form a different types of peptide bonds and gives a long chain polypeptides so as to form a protein. That's why alpha amino acids are acting as various kinds of precursors or a building block to develop various types of protein in the nature. This is the beauty of alpha amino acid. This is the smallest unit which is continuously repeated by formation of peptide bonding, and they give formation of such type of polypeptides in proteins. So this is very important property, that is alpha amino acids gives formation of polypeptide type of linkage. And how it happens? NH2 group of one alpha amino acid combines with carboxylic group of same or another type of amino acid of second molecule, then so as to form various kinds of proteins, and this is important property given by the various type of particular nature. So these are the chemical properties shown by various kinds of alpha amino acid. I will again repeat. These are amphoteric in nature, showing both groups, negative, positive groups. Zwiter ion formation, that is neutral in solution. Alpha amino acid contains NH3 plus charge and CO minus group. Same species containing positive and negative charges, known as Zwiter ion formation. This property is shown by alpha amino acid. Then it is showing specific isoelectric point, the specific pH at which amino acid having no net charges, not positively charged, negatively charged, and this property is shown by various types of alpha amino acid. It is also having buffering capacity, formation of peptide bonds of amino acid so as to give a polypeptides, and such linkages are observed in protein. So these are the chemical properties of amino acids. Now we move towards physical properties. Now the important physical property first, that is the solubility of alpha amino acid. Most of the alpha amino acid, as we are getting these alpha amino acid from living sources, vegetables, food materials, they are having mostly soluble in water, while some of the alpha amino acid which are insoluble in non-polar solvent. So in water they are majority soluble, while insoluble in non-polar solvent system, that is particularly benzene, NHane, cyclohexane, all such kinds of non-polar solvents, they does not show solubility with alpha amino acid. Second, melting points. Most of the alpha amino acids are having higher melting point, greater stability, decomposition points due to ionic structure. They decompose only by formation of Zwiter ion or ionic structure. This is very important. Means melting point is higher, decomposes only by formation of Zwiter ion. Third, majority alpha amino acids are, if they are existing solid form, mostly solid form, that is crystalline in nature. They exist in crystalline solid at room temperature. Optical activity, polarized light, no doubt, except glycine, all of the alpha amino acids are having alpha carbon atom as a asymmetric center, and as it is having asymmetric center, very important property, all except glycine are the optically active compounds containing at least one asymmetric center. So they rotate plane polarized light and they are optically active. So these are the some important physical properties of alpha amino acids. Alpha amino acid. So such kind of properties, physical properties, are shown by the alpha amino acid: soluble in water, having higher melting points, crystalline in nature, they are optically active, as well as rotate plane polarized light. This is about the particularly physical property. Now we have to study the most important chemical property, that is the isoelectric point. What is isoelectric point? Particularly shown by alpha amino acid. Now see the slide. Isoelectric point means what? Whenever the solution of alpha amino acid is prepared in water and placed in a simple electrochemical cell, as alpha amino acids are soluble in water, they are exist, no doubt they are existing in which form? That is Zwiter ion form. And one of the experiment we are going to conduct, that is voltage is applied to this electrochemical cell and we are going to add some of the acid to the solution. What happens? As we add acid to that particular solutions, the Zwiter ion start to convert either positively charged cation or negatively charged cation. If it is converted into positively charged cation, whenever the pH is lower than isoelectric point, means first we see isoelectric point is nothing but a specific pH at which the amino acid exists as a Zwiter ion. It is having no net electrical charge, not positive charge or negative charge. and therefore if it is having no specific single charge, positive charge or negative charge, it is neither attracted towards anode or it is neither attracted towards cathode. Such kind of specific pH point at which there is no migration of amino acids towards positively charged electrode or towards negatively charged electrode. Such kind of pH is known as isoelectric point. Listen carefully. Isoelectric point means what? First, see the difference. Suppose we are taking a solution of amino acid. No doubt it is existing in which form? Zwiter ion form. NH3 plus CO minus. Whenever we are going to adjust the pH of the solution, okay, if we are adjusting pH towards lower, means we are going to add acid. If we have to add acid in the Zwiter ion form, COO minus group is get converted into COOH. That is means low pH, and we are getting NH3 plus charge at a net charge. If the alpha amino acid, see the left side in the screen, alpha amino acid containing NH3 plus charge, at that time this positively charged alpha amino acid is get attracted towards negatively charged anode of the electrochemical cell. The solution is taken in electrochemical cell. Means if it is having positive charge, it is attracted to negatively charged anode, a negatively charged electrode, and if it is having negative charge means higher pH in the base. If it is having a negative charge in alpha amino acid, see the structure of right side, it is having negative charge, get attracted towards what? Positively charged electrode of the electrochemical cell. It happens whenever a normal electroanalysis is done, but whenever a specific pH comes, at that time alpha amino acid is existing in the form of Zwiter ion, means it is containing positive charge on NH3 group and a negative charge on COO minus. It is having both structures. At that pH, this Zwiter ion neither be attracted towards positively charged electrode nor towards negatively charged electrode, and it remains stable. This point at which a specific pH at which amino acid having no net charges, not perfectly positive or perfectly negative, this point is called as isoelectric point, and this is specific for each alpha amino acid. For alanine, serine, proline, all alpha amino acids are having specific isoelectric point. It is similar property to viscosity, melting point, boiling point. Like this, all kinds of alpha amino acids are having specific isoelectric point, and this is a special property which is shown by these alpha amino acids. So the important is what is isoelectric point and how it is conducted. This experiment is conducted. So remember this important point. What is the isoelectric point of alpha amino acid? The pH at which there is no any net charge on alpha amino acid, and it is neither attracted towards negatively charged electrode nor attracted towards positively charged electrode, and having zero net charge. Such kind of pH is called as isoelectric point of alpha amino acid. So in this short video we have studied physical property of alpha amino acid, chemical property of alpha amino acid, and isoelectric point. The important point is the what is isoelectric point of amino acid. Remember the chemical properties, physical properties, and all these properties of the alpha amino. Okay, we will see the rest of the part in another lecture.